Now, back to Krebs cycle. It's the circle of life. And the link below is to the Lion King video. It can be easily broken down into two parts. It's the decarboxylation part of the cycle, and then the recovery part of the cycle. So oxidative decarboxylation means that the carbon molecules of acetyl-CoA, or of the acetyl part of acetyl-CoA, are oxidized to carbon dioxide. Oxidative decarboxylation. And oxidative regeneration implies that the participants of the cycle have to be regenerated back to be competent to take up another pair of acetyl, another acetyl molecule, another pair of carbons, and a molecule that's competent to react with two acetyl molecules as oxaloacetate. So the rest of the cycle is there to recover oxaloacetate for the next circle of the cycle. Now, energy is harvested from the cycle both during the oxidative decarboxylation steps and the oxidative regeneration steps. So NADH is produced here and here, as well as from malate to oxaloacetate. Additionally, FADH2 and GTP are also produced. So, in order to introduce you guys to the citric acid cycle, I decided to make the happy fun citric acid cycle diagram. Because the intermediates of the citric acid cycle have perplexing names like succinate or fumarate, I figured I'd actually show you why these intermediates were named that way, and hopefully make the citric acid cycle less dreary, because it's very important to understand. Now, happy fun acetyl-CoA comes in, and here the two blue circles are the two carbon atoms that will be lost during the citric acid cycle. And we will talk a little bit about when the actual carbon atoms that come in are lost in the next few slides. And there's a condensation reaction happening with one of the intermediates of the citric acid cycle called oxaloacetate. Oxaloacetate is an acetate of the oxalic acid. Oxalic acid is an acid that gives a sour taste to a lot of fruit. It's actually named after wood sorrel or oxalis, those little four-clover plants that you can sometimes see growing in warmer climates in Laramie. But it's also very enriched in raspberries. So the taste of raspberries is oxaloacetate. And when these two things condense and CoA leaves, you form citrate, which is a defining flavor of oranges and lemons. Citrate is actually citric acid because of the environment. All of these intermediates of the citric acid cycle are acids, or so they're monodicarboxylic acids. There's multiple carboxylic groups in some of them, and because of the environment of them inside the matrix of the mitochondria, they're in their deprotonated form, so they're 8s. Remember how amino acids were 8s as well, right? Glutamate versus glutamic acid. So citrate is actually citric acid deprotonated. So we make citrate via carbon-carbon bond formation or condensation of oxaloacetate and acetyl-CoA. Then we isomerize it, moving one of the methyl groups over via dehydration-rehydration reaction to form isocitrate, and that is catalyzed by an enzyme called aconitase, because citrate, to get to isocitrate, goes through a cis-aconitate isomer, something that was dropped in a modernized citric acid cycle. Now, the next step is to make some NADH and lose one of the carbons, and isocitrate is decarboxylated by isocitrate dehydrogenase and NADH, happy NADH, and carbon dioxide are produced, and we form alpha-ketoglutarate. Now, that sounds like an extremely convoluted name, but what it is glutarate, which is glutaric acid, with a keto group in the alpha position. Hopefully for everybody who took organic chemistry, which should be all of you, it becomes a little bit easier to decipher why this compound is called alpha-ketoglutarate. Glutarate is a close relative to glutamate. In fact, you use alpha-ketoglutarate to make glutamate, which is an amino acid, and glutamate is the main flavor of savory umami taste that you get from chicken soup. Next step is another oxidative decarboxylation to produce an ADH. These are the two big decarboxylation steps. These are the two decarboxylation steps in the citric acid cycle. This is the two places where carbon is lost. So here we form another NADH, and the second carbon leaves. And what we end up here is succinyl-CoA. The reason succinyl-CoA is here an amber brooch is because succinate is actually named after amber, the semi-precious gemstone, and because there's a CoA in it, I just came up with that mnemonic. It's being held by something pretty like coenzyme A. It's being presented. So this completes our oxidative decarboxylation half of the cycle, and now we're trying to regenerate oxaloacetate right here. So CoA leaves, and we have a substrate level phosphorylation occurring, forming GTP and just succinate, or succinic acid, or actually spirit of amber, which is the old-fashioned name for succinic acid. Because when succinic acid or succinate was discovered, it was actually distilled from amber. Again, substrate level phosphorylation to give GTP and release CoA, and then GTP can be easily converted to ATP by just moving phosphates around. So it's a source of substrate level phosphorylation of ATP in this citric acid cycle. Next is conversion of succinate to fumarate. It's another dehydrogenation reaction which allows us to reduce FADH to FADH2, which heads off to the electron transport chain, just like NADHs will. And actually, the conversion of succinate to fumarate, the enzyme that catalyzes it, directly feeds FADH into the electron transport chain because it's part of the membrane of one of the complexes, which we will see tomorrow. Fumarate is an intermediate of the citric acid cycle. It actually has a really strong fruity flavor. That's why it was named for fumarine or fumare, something like that. It's a plant. Sorry, but fumarate is also the smell of mushrooms. It's the fruity smell that mushrooms have, and it's produced on grand scale for the food industry because it will give the fruity taste to chewing gum and all kinds of other products you buy in the grocery store. And then after hydration, fumarate, hydration is the addition of water. After a hydration reaction, fumarate is converted to malate, which is named after apples, and is very prevalent in apple fruit. Malic acid, taste of apples. This is the final conversion of malate to oxaloacetate. Completes the cycle and is the final dehydrogenation reaction done by malate dehydrogenase that gives off another NADH and recovers oxaloacetate. The amount of oxaloacetate in the mitochondria is actually one of the limiting factors for how fast the citric acid cycle can perform all of the reactions described here. So also, if you guys remember from fatty acid oxidation and actually not from fatty acid oxidation, from gluconeogenesis, malate oxaloacetate shuttle is how you can get pyruvate out of the, how you get oxaloacetate out of the mitochondria to allow for further gluconeogenesis steps in the cytoplasm. And this is a summary of the steps, of the 8 steps of the citric acid cycle. Again, first we make citrate by forming a carbon-carbon bond between acetyl part of acetyl-CoA and oxaloacetate. Then we isomerize citrate to form isocitrate. Then we start harvesting NADHs and releasing carbon from the molecule, ending up with succinyl-CoA, which is then converted to succinate, fumarate, malate, and oxaloacetate during the recovery phase of the citric acid cycle. And since people feel the need to learn the names of the citric acid cycle intermediates, here are the 2 mnemonics. If you have more mnemonics, I will give you a point for mnemonics for the citric acid cycle, but they need to be, but if I know that you got just copy pasted it from the internet, I will not give you a point. But the 2 that are easy to memorize is, can I kiss some sexy French man? Okay. And this is courtesy of Rita Fatima, which is one of the students I taught in the past. And the common one is, can I keep selling sex for money, officer?